The items that you're going to need for your seed lab is your science journal, open to that page with the bean and the corn, your lab sheet with your name on it and a pencil, and then your lab materials, magnifying glass, the 4 kinds of seeds, which are the bean and the corn. Those are very important, those 2. Then the extra ones you could do are the sunflower and and the pumpkin. And then you'll need a toothpick also. And colored pencils. Don't forget about your colored pencils because that'll help you.
All right. Today we're going to start with our corn seed, or our monocot. This is a monocotyledon. That means it has 1 seed leaf. So it's not going to split in 2. There is a seed coat or testa around the outside of your seed. These seeds have been imbibed. That's our word that we learned this week in science. But we're picking off that seed coat. And that will start to come off. It's almost going to look like the hull of the, of the corn. And that's the part that sometimes when you're eating popcorn, it gets stuck in your teeth. We've all kind of been familiar with that. So get some of that off. And then we're going to move to, there's a piece of it. We can see it there. Just thin little coating on the outside. Normally that would be so hard it wouldn't come off. When we look at the corn, we can begin to see the embryo in our corn, and that's this yellow part the white part right here. The top part of that, where it meets the yellow, is the plumule, the part that's going to become the shoot of the plant, and then the root is the radicle that's down here. This yellow around the embryo is that food storage or food tissue, that is the endosperm. So now we can move to our bean.
Before
we move to our bean, we're going to take a look at how to draw that. So we want to start to make our corn seed so we can label all the parts. And then I'm going to move ahead and show you what that should look like when it's done.
Notice all the labels. Science words are a level 4 work.
So we're having all the science words along with the regular terms. Yeah. Everything's colored correctly. We can even see that testa or seed coat that we pulled off in the background here.
And a sentence to explain what we saw. Could you read the sentence?
The corn seed is a monocot. I found the embryo and the testa. And then we didn't say it either, but we need to make sure that we have our name and our room number at the top of our lab sheet. Okay. The second seed that I'm going to do with you is a dicot. This is our bean seed, and these also have been imbibed. So this is like I said, it's a dicot. There's 2 parts to this, and if we are very careful, we can begin to split this. So I'm just kind of sticking my fingernails in here, and I want to get that seed coat off. And you can see this as we pull it off.
It's really thin too.
Yeah, and it's going to come off easier than the corn did. Okay, so here it is. That's the
seed coat.
The seed coat.
That was the testa.
That was what was protecting the seed before it was imbibed. It was hard. And here we can see the 2 parts. This is why it is a dicot. It has those 2 seed leaves. We're going to also look at the embryo here, and I'm going to very carefully, I should probably use my toothpick. There it is. This is that baby embryo. I just lost the shoot of it. And this part is the root. So these were actually together, like that. And you can, if you look really closely, you can actually see where those leaves would begin to form. This is the root that would go into the ground. This is the shoot that would then become the leaves or the top part of the plant.
Can you point out the root again?
This is the root, which is also called the radicle. That's the science.
Can you point out the shoot?
And this is the shoot or the plumule.
Okay.
These are our 2 seed leaves, which are the
That makes it,
Cotyledons. So this is a dicot, dicotyledon. And this is our testa, our seed coat. So we're going to move to the,
once it's dissected, we go back to our lab sheet.
we want to draw our parts here, making sure that we draw the 2 seed leaves. We can add in that embryo, and we would want to draw the testa here and make our observation. And we can see here we have the science words, we have the regular language for what we call those. In the background here we have that seed coat or testa, and then we have our 2 parts of the dicot plus the embryo here, and the parts of that are all labeled. I observed that the bean split in 2. This makes it a dicot. So sentences at the end for our observation.
So this is the 2 seeds that are required. Then to go above and beyond, you could add dissecting 2 other seeds. The sunflower, which is the black one, and the pumpkin.
This is your sunflower whole. So you would need to split this in 2. And when we break that, we end up with the seed coat, which is just the empty shell. Most of us have had sunflower seeds at some point. And this sunflower seed then breaks in 2. And this would be 1 part of that dicot. So 1/2 of it. If we look at the pumpkin seed.
And then I would go to my sheet and label all of those
Right,
again.
oops, sorry.
Yep,
Yep.
no.
This would be the sheet, and this is where you would put all the parts of your sunflower seed, noting that it has 2 seed leaves. It is a dicot. Then we can take our pumpkin seed, and this also would split in 2. You can use your fingernails for that. And we're going to end up with the testa, the seed coat that's split in 2, and then also our pumpkin seed inside. If we look closely, this little nub on the end is going to be your embryo. Right here. And that breaks off real easily, so you really have to look, but you can kind of see where the shoots would come up, and this little pointed part, that root's going to come down. The rest of this is all the food storage, and this also will split in 2. It's going to break, so you got to be kind of careful about it, but you can see that it splits in 2 parts. If you're very careful, you might be able to get 2 whole parts. Mine kind of broke apart here, but into the 2 seed leaves then. So then again, you would come back to that lab sheet, and you would draw the parts of that and label them.
And color.
And color, yep.
All right.
And we can see too on the sunflower seed how you're going to need to add some stripes and gray, and we really need to get detailed to get that 3/4.